Hi everyone. So today I'll be talking about how we prepare to switch between responding. So how we prepare to switch between responding via our hands or via our mouth. And what I'm going to show today is that the preparatory dynamics that we use operate in such a way like traffic signals on the road. And that's going to hopefully be more clear later on. But the general question I'm interested in is, how we control our attention. And this idea of this experiment that looks at how we prepare to switch between different modes of responding is one way that we're looking at how we efficiently control our attention and switch from one task to another. So first of all, why is this important? Why are people studying this? And it probably comes as no surprise that every day we have to switch between tasks. So we have to focus on this project and then switch our focus to another, or we could be working and then suddenly have to do something that's outside of work. And not surprisingly, there have been a lot of studies, especially in psychology, that have looked at task switching and how we can efficiently overcome or prepare when we switch from one task to another. Not a lot, though, has been done on what I'm going to be talking about today, which is response switching. And I feel like this is incredibly important in our increasingly multimodal world where we juggle between responding in certain ways. So if you think about a pilot in a cockpit, he has to issue commands either verbally or manually, or even when you're on the phone with customer service or you're Skyping, you're having to switch between responding with your voice or responding with your hands. And in some cases, it's the millisecond difference in the efficiency that we have in switching can make a big difference. So this is an example of what a typical switching experiment in psychology looks like. And this paradigm that I'm showing you right now is similar to the one that I used in the experiment I'll talk about today. So in a switching experiment, participants complete a series of trials. So they do a task for a number of trials. And here I'm showing you a sequence of 2 trials. This is the first and the second over there. So in this experiment, they have to switch between responding with their hand or responding with their voice, so vocally. When they see an H, at the beginning of the trial, they receive a Q, and the Q can either be an H or a V. If the Q is an H, at the end of the trial, they have to respond using their hands. But if the Q is a V, at the end of the trial, they have to respond verbally or using their voice. So at the beginning of the trial, they're told how to respond. And then after that is a delay period where they can prepare for the response that's going to come up. After that delay or preparation period, a stimulus comes on the screen. The stimulus can either be an L or an R. If it's an L, they have to respond left. If it's an R, they have to respond right. And how they respond depends on the cue that they're giving at the very start of the trial. So in this trial right here, they're given an H. That means they have to respond with their hand. So if they see an L, which stands for left, they have to respond by pressing a button with their left hand. Now in this next trial, they receive a V as a cue. And at the end of the trial, they receive an R, so they have to respond right. And because V is the cue, they have to respond verbally or using their voice. So participants complete a sequence of trials like this. And in the sequence that I'm showing you, what you're seeing is a switch trial, where they switch from having to respond with their hand to having to respond with their voice. But note that participants don't know what the upcoming trial is. It can either be a switch trial like this, where they switch from hand to voice, or it could be a repeat trial where they do hand, and then the next trial they do another hand trial. So what do we know about task switching? So in experiments such as this, what they find is that there are costs to switching, which means that when you have to switch, your performance on this particular trial tends to be worse than if you did not have to switch. So on switch trials, what people typically find is that participants create more errors, and participants are slower to respond because they have to reconfigure and switch to the new task. And these effects seem to persist even when you give participants time to prepare, like in that paradigm I previously showed. But it probably varies depending on what task you have to switch to. However, there is evidence that we can be more efficient in preparing to switch. So obviously, if you're given a cue and if you're given time to prepare, you can better overcome switch costs, you can better prepare and control your attention. And this control of attention is critically been linked to activation of certain regions in the brain called the frontoparietal network. So briefly, with the frontoparietal network, this idea of a frontoparietal network came about studies that found co-activation of the frontal area of the brain and the parietal or top back area of the brain. And they find that these areas activate at about the same time. And critically, they found that these areas activated during conditions that demand a high control of attention. So they're more active if you have to focus in the face of distraction. And in switching paradigms, they've seen that these regions are more active on switch trials compared to repeat trials. So when investigating how people prepare, they also found that it's not only these regions that are active. They also found that regions that are important for executing the task or for processing the stimulus that's about to come up are activated even in the absence of that stimulus and even before that action is executed. So what people seem to find is priming of regions that are important for the task that's about to be executed. So in these two images that I'm showing over here, in the first image I'm showing you sensory areas that are active when a stimulus is on the screen. And in the second image, what I'm showing are these same areas, but are now also active only when there's a cue that indicates that this type of stimulus is going to appear. So the same areas that are active when the stimulus is on the screen are also active when there's a cue that's indicating these kinds of stimuli. What's also interesting is how this type of activation is modulated by switch or control demands. And what they find is that in just preparation or in priming areas that are important for the task that's about to come up, priming or activation in these areas is even greater when it's a switch trial or when the condition is when the condition demands high control. So the priming seems to be greater in the face of distraction and when you really need to focus or concentrate. So what I'm interested in today and what this experiment that I'll talk about is going to try to address is the relationship between these control mechanisms that I talked about in frontoparietal cortex and how they relate to mechanisms in task-specific regions. So how do the control regions relate to the processes in task-specific regions that are important for executing the task? And what we think is happening is that these control regions, like the region in frontal cortex, is talking to these task-specific regions and modulating their response in order to prepare for the upcoming task. The way that we study this is by using lag cross correlations. So what we do is we take activity from a certain area of the brain and then correlate it with activity in other areas of the brain. But what we do is also include a timing factor. So we not only know how this brain area correlates with this brain area when they're aligned in time, but we also know whether or not activity in a certain area can predict activity in another area at a later point in time. So just to show you a graph of what the analysis looks like, we can have these two different waveforms. And if we look at their correlation when they're aligned right in time, there's a relationship, but we may not be able to see a relationship because the relationship only evolves later on in the period. So what we do in lag correlations is we shift one waveform relative in time to another to better identify whether there's a correspondence in activity in these two different regions, but may be happening at different time points. So what lag cross correlations allow us to do is to identify the correlation of activity between regions of the brain and also infer the relative order of activation. So we have some idea of whether or not activity in this region precedes or may even predict activity in another region of the brain. So for example, in this experiment, what we might see is that you have activity and activation in frontal regions, and then later on, you'll see that this activity can predict activity in other regions of the brain important for the task, like say motor regions when you have to execute a manual response, or speech preparation regions when you have to execute a vocal or verbal response. So cross correlations can also be powerful because they can detect patterns of activity. And in typical neuroimaging and cognitive neuroscience experiments, you have the case where activity is lined across the same time points. So if you have subjects that start processes at different times, that activity may not sum up and it may get washed out or not easily detected. Whereas for cross correlations, it doesn't matter as much where the activity starts as long as there's some consistent pattern. So for neuroimagers in the audience, you can think about it as the idea of functional connectivity, but with timing information or lag included. So we use lag cross correlations using this method called event-related optical signal, or EROS, which is a form of optical imaging. And EROS contains high spatial and temporal resolution. So we can not only identify where activity is coming from, but when it happens. So optical imaging, briefly, is where we use near-infrared light. So we shine near-infrared light onto the scalp, and a detector nearby reads or measures the light that comes back out. So you have several sources that shine light and detectors that detect light positioned at different parts of cortex, and they're positioned in such a way that, which source comes on at a certain point of time is positioned in such a way that we can identify when we're at the detector which source that activity we're getting must be coming from. So the main idea behind EROS is that it measures the changes in optical scattering of neural tissue. So we know that there's a difference in the optical properties of neural tissue when it's active versus when it's not. When it's active, it becomes more transparent, so more light is able to travel down the cortex, and it takes longer for it to come back out. So our main measure for optical imaging in terms of EROS is photon delay, where we compare how long it takes light to come back out in one condition versus another. So just to refresh your memory, we use this in the context of this experiment where we have cues at the beginning telling people how to respond. At the end, they get a stimulus and they respond based on the cue that they were given for that trial. And what we're interested in looking at is this preparatory period. So this time after the onset of the stimulus, where we want to know what subjects are doing in order to prepare for the response. And what we're most interested in is the interaction of these control mechanisms that we think is instantiated in frontal cortex, and how they modulate the mechanisms in those regions that are important for actually doing the task. In terms of behavior, what we see is that we have this usual effect where participants are less accurate or create more errors in switch trials compared to repeat trials. But in terms of reaction time or how fast to respond, when we compare switch and repeat trials, the effect is only marginal. And it might be because the task that we're using is easy. We're just switching between hand response and mouth response, and not between two totally different tasks. But it might also be because we give them this preparatory period, and they in fact utilize this preparatory period and recruit these control mechanisms in order to prepare for the task. So here what I'm showing you in red are regions that are more active on switch compared to repeat trials. And we confirm the findings of previous literature where we see greater activity in parietal cortex and also greater activity in frontal regions of the brain right here. We also look to see whether or not we have what the effects that we expect of increased activation in areas that are important for doing the task, even though the participant isn't doing it yet. And here we found the typical priming effects, where in red, I'm showing regions that are greater when participants have to prepare for a vocal response versus when they have to prepare for a manual response. So in red right here are regions in the left hemisphere of the brain that in previous literature has been shown to be important for speech production. So here we have Broca's area, and then here we have the mouth area of the motor cortex. And similarly, when we look at manual to vocal, in blue are regions that are greater for manual responses versus vocal responses. And here we see priming in the hand area of motor cortex. But again, what I'm most interested in is how the frontal control mechanisms interact with these task-specific response mechanisms. And to do that, what I did was I took the activity in the frontal cortex in the previous slide that I showed earlier, and I correlated it with the switch effect in the vocal condition and in the manual condition. So what I'm showing along the x-axis are different lags. And in blue are areas that are negatively correlated with this effect right here in right frontal cortex, separately for vocal and manual. And in red, you'll see the areas that are positively correlated. So negatively correlated areas you can think of as areas that are downregulated with respect to right frontal areas. And areas in red are areas that you think of as being upregulated in response to activity in this area of the brain. So what you find, let's say for the vocal condition, is downregulation of a region in the right motor cortex, and particularly in the region of the hand area. And we know that this hand area is the irrelevant region for the vocal task. Similarly, for manual, what we're seeing are the blue regions in areas that are important for speech production, which are irrelevant for the manual task. So it's as if when this right frontal cortex is active, it then leads to downregulation of regions that are not important for the current trial, but were important previously. And then in later lags, what we see is that for the vocal condition, we see upregulation of regions that are important for vocal or speech production. Similarly, for manual, we see activation of regions that are important for responding with your hand, which include the hand area of motor cortex. So here, just to summarize, what we see is upregulation and downregulation of activity, but first downregulation, then upregulation in response to activity in this frontal area. So just to summarize, here what we see is that connectivity between frontal control regions and task-specific regions seem to be important for preparation, and that it proceeds in this traffic signals kind of way where you stop flow in one lane and then you start flow in another, where first you stop or downregulate activity in the regions that are not relevant for that trial, and then upregulate activity in regions that are now relevant for that trial. And future directions in this experiment include looking at similar paradigms like this and also examining the importance of structural connectivity. So how important is the structural connection, like the white matter between these regions? How does that support performance? And we're also interested in looking at age-related changes because we know that switching is one ability that grows especially more difficult with age. And we're also interested in applying this to augmented cognition, where we can monitor people's states, we can monitor how prepared they are, and then we can adjust or help their performance by providing feedback on how they're doing. So I'd like to thank these people who helped a lot in this project: my mentor, Kathy Lowe, my advisors, Monica Fabiani, Gabriele Guton, Art Kramer, and Ed Macklin, the Cognitive Imaging Lab, who helped a lot with this project too.